Good morning, guys. So you have gotten through modules 1 and modules 2 of your DNA invitation. Yay, you're halfway there. We're gonna go through module 3 now. So module 3 has some notes that are due. I want you to make sure you take really good notes. This is a really important module. That'll help supplement some stuff that you might not have gotten in class. Hopefully that you've gone through module 1 and you've watched the recap video, and you've gone through module 2 and you've watched all those videos and done those assignments. Module 2, you had to do a KWL chart. So make sure that you are completing those and that you are getting those done. Okay, remember that these are all due before the unit test, like the day before at 11:59. These are intended to help you learn and prepare for the unit test, not as something to do after you take the unit test. These assignments will be locked on Canvas, and nothing will be accepted that is dated after the unit test. Okay, I will be collecting the invitations turn-in tray before your class takes the test. So make sure everything is in the tray before you go in to take your tests. That way you get points. If you are absent that day, or if you have any extraordinary circumstances where something's happened, please just send me an email, let me know, and I will be more than happy to extend that deadline for you, but it will be on a case-by-case basis. So let's go ahead and get started with module 3. Module 3, how does DNA make proteins? So these are some questions I just want you to think about as we're going through the notes. Why does DNA need to be replicated? Why do we need to replicate our DNA? Why can't we just use the original copy we have? And then why does DNA replication take place? Okay, why does it take place? And when does it take place? When does DNA take place? When does this go occurring? Why is it occurring and when is it occurring? Okay, so DNA replication. Replication is the process of copying DNA during synthesis. So the interphase portion of the cell cycle. Okay, so this is when the cell is replicating its DNA, getting prepared to divide, right? Replication assures that every single cell in your body has the complete set of the same genetic information. So the cells in your eyes have the same genetic makeup as the cells in your fingers, right? What's making them express differently is what genes are turned on, what exons are being expressed. So as you can see here, we have the DNA replication fork. So this is the strand that is getting replicated, and this is the strand that is not getting replicated, okay? So it's getting replicated this way, okay? DNA is unzipping as it goes across the DNA molecule. This strand is getting replicated, and this strand is not. It's lagging. It's not getting replicated continuously. It's creating these Okazaki fragments, right? As it's replicating. So we're going to talk about replication in prokaryotes now. It's a little different because remember that prokaryotes have the circular DNA, right? So in prokaryotic cells, DNA begins at a single point in the chromosome and then proceeds often like this in two directions until the entire chromosome is replicated. Okay, so it starts here at the initiator. Okay, oftentimes you'll have two helicases that are unzipping the genome in opposite directions. Okay, and you have two different replication forks. Okay, two DNA polymerases are replicating opposite fragments as it goes around. Okay, the end of replication, you'll have two completely different sets of DNA. One will not completely different, identical sets of DNA, but two separate whole double helix sets. Okay, so DNA structure helps explain how it duplicates. Hydrogen bonds between two strands are easily broken. So the hydrogen bonds here between the C and the G and the T and the A, okay, they're easily snapped, they're easily broken, okay? Each single strand then serves as a template for the new strand, okay? So the bonds are going like this, and then as soon as they're done being replicated, they're just sewn back together, okay? So there's like a little bubble that you are getting replicated in before it's sewn back together. Okay? So, DNA replication. In humans, our DNA replication is semi-conservative. That means that the parent strands of the molecule, so the original DNA molecules, okay, they remain intact. The new DNA molecules are half of the old DNA, half new DNA. So half of what wasn't replicated and half what was replicated, okay? So, replication. Here are the steps of replication. DNA helicase, they're enzymes. unzip the DNA at several places along the strand by breaking the hydrogen bonds between the base pairs, okay? Once the strands are separated, helix destabilizing proteins, so something that's going to shake up the protein, the helix, and make them separate, bind to the single strand, preventing the strands from coming back together, okay? Floating nucleotides pair up with the bases on the template strands as they are unzipped. Okay? DNA polymerase catalyzes, makes it go faster, and links the nucleotides together to form new strands that are complementary. Okay? Two identical molecules of DNA are made. Each DNA molecule has one old strand and one new strand. Okay? Old and new are now a new helix. Old and new are now a new helix. Okay? I want you to watch this video on your own. We're not going to play it since we're just trying to get through the notes. But make sure that you watch this video and that you really get to know and get familiar with the process of the structure of DNA and the replication. Okay? It's just Hank Green. We love Hank Green. Anyways, DNA is anti-parallel. So that means that two chains are running in opposite directions. Okay? Replication always occurs in a 5' to 3' direction, okay? The 5' is the phosphate group and the 3' is the sugar group. So over here, we see that this is the 3', okay, the sugar group, and this is the 5'. So it's going 3' to 5' here. And here it's going 5' to 3'. They're running opposite ways, okay? So replication always occurs 5' to 3'. So it always goes this way. And then if we were to do replication on this other strand, it would go this way. Okay? Leading and lagging strands. This is also really important as to why DNA is going to be synthesized continuously and the other one is not. Okay, so this 5' 3' direction requires one DNA strand to be synthesized continuously and the other to be synthesized discontinuously. Okay? So the leading strand, the one that's going 5' or 3' already, is being continuously replicated. It doesn't stop. There's no stopping until it reaches the end point. The lagging strand is the one that goes 3' to 5'. And it's discontinuous replication. So it's replicating in short bursts, okay? That produces Okazaki fragments, which will eventually be joined together by DNA ligase to create one entire new DNA strand, okay? So this is a really good picture of how replication works. Okay, so this is DNA helicase. Okay, DNA helicase is unzipping the genes as it goes this way. You can see that it's going on this side, the 5' to the 3' direction. Okay, and this one is going. Okay, you can see as it's going, it's unzipping. This is the leading strand up here. This is the lagging strand. Okay, so it's unzipping the genes. It's being replicated, okay, by DNA polymerase. It's getting these complementary nucleotides, okay, that are being attached to this other one to create a brand new strand of DNA, brand new helix of DNA, okay? And this side, DNA polymerase is doing the exact same thing, okay, but instead of doing it in these continuous motion, right, as we see here, it's all one motion. We see it in these chunks. Okay, do you see how there's a gap here? These are free nucleotides. Okay, they're making these jumps that are eventually going to be sewn together to create one long continuous DNA strand. Okay, so replication is really, really fast. So I know that I am talking fast. Replication is faster than this. Okay, and this is because replication happens so frequently, it has to be really fast. Okay, in humans, 50 nucleotides are added every second. So in the time of this video, I'll do the math in the caption of the video. But in this video, there are probably going to be over 1,000 nucleotides made. Just think about that for a second. In the time it's taken you to watch this video and to take these notes, you've made over 1,000 nucleotides in your whole body. Okay. So DNA polymerase is not just sewing it together. DNA polymerase is going to detect errors in the DNA strand in replication. Replication is not a perfect process. So DNA polymerase is the first step to catching those errors and replacing them with the correct nucleotide. So it takes out the incorrect ones and puts the correct ones in there, okay? Okay, so replication is done. So protein synthesis can happen, okay? When a protein is needed, the cell makes a protein through protein synthesis, okay? This is the process of getting the information from the DNA strand, sending it through 2 different processes that we're going to go through, and then creating a protein from that tells our body what to do. Protein synthesis must occur in the ribosome, which are located in the cytoplasm. Therefore, the code must be carried from the nucleus to the cytoplasm. So we're going to talk a lot about the central dogma of biology. Okay, so the central dogma is is a theory, not a theory, it's been proven, but just it's a term for describing the flow of information from DNA to RNA to proteins. So replication copies DNA. Transcription converts DNA to RNA. Translation interprets RNA into a string of amino acids or proteins. So the way I think about it is replication is copying DNA. Transcription is like you're taking notes. DNA is taking notes and putting it into a shorthand, like the court reporters do in the courtroom, to then be translated into something else later. So it's like you're taking notes in class, maybe for somebody, and then later you're going to go and you're going to translate them into your own words or maybe into another language, and then it'll get passed on to the final product or the last person, if that makes sense. So RNA, what is RNA? We've talked a lot about DNA. Now we're going to talk about RNA. RNA, ribonucleic acid. Remember that DNA is deoxyribonucleic acid. Okay, so RNA is ribonucleic acid, so a chain of nucleotides. It's not a double helix, it's a single helix. Okay, it acts as the middleman between DNA and then in the nucleus and proteins in cytoplasm. Okay, DNA and eukaryotic cells cannot leave the nucleus. RNA can. So RNA leaves the nucleus and then goes through transcription and translation, and becomes proteins in the cytoplasm. Okay. Another important thing that we need to know about RNA is that the nucleobases, the nucleotides, are different. So in RNA, C and G are still pairing up. They're still besties. However, with A and T and A, they're not besties anymore. They broke apart. Now A is besties with U. Okay, A and U are besties. Adenine and uracil are besties. And thymine dropped off the face of the earth. Thymine is not there anymore. Thymine transferred to a different state. Okay, so let's talk about the differences between DNA and RNA really quickly. Okay, so DNA on the left, deoxyribonucleic acid, double-stranded sugar phosphate. Okay, deoxyribose. Okay, seeing how this is going. Something that's really important to note is there's an alcohol pairing here and there's a hydrogen here, and then when you go over to ribose, there's 2 alcohols and not a hydrogen here. Okay, in DNA we talked about thymine. Okay, thymine and uracil are enemies. They are enemies because they are so similar and they both want to match with adenine. Okay, and down here at the bottom we have the common nucleobases. Okay, this is a really good picture for just thinking about what the differences between DNA and RNA are. I encourage you to reference this as you're studying. So RNA molecules. There are 3 important types of RNA molecules. There's actually a lot more types. There's, I think, probably hundreds of types of RNA molecules that we're going to not discuss in this class, but they're the 3 ones that you need to know for this class. Okay, messenger RNA, or sometimes known as mRNA. Middle message that is the translated form of a protein. Okay, so this is what mRNA looks like. It is just a strand of RNA. Ribosomal RNA, or what form the ribosomes. Okay, we talked about ribosomes when we were talking about cellular organelle, membrane-bound organelle inside eukaryotic cells. This is a ribosome. Okay, this is actually a really big ribosome, but this is what a ribosome looks like. It's made up of rRNA. Okay, and then tRNA is transfer RNA. So it's bringing the amino acids from the cytoplasm to the ribosome so that way it can be processed into a protein. Okay, and it has this weird shaped structure so that way it can carry 3 base pairs at a time. Okay, so transcription. Transcription is the process of copying a DNA sequence to produce a complementary RNA. Okay, a really important enzyme that you need to know is RNA polymerase. It is the enzyme that bonds the nucleotides together to make RNA, okay? So here we go. Here's a really pretty picture that I found. So as you can see, the DNA molecule is unwinding. You still have the double helix. Here's the strand, here's the strand. As it's unwinding, RNA polymerase is going down the strand, okay? And it's creating these complementary base pairs to this top strand, this template strand of DNA, okay? As it's doing that, the mRNA is leaving the nucleus. It's creating mRNA here. It's leaving the nucleus to enter into the cytoplasm through the nuclear pore, okay? So RNA polymerase is creating the nucleotides. It's creating the mRNA strand. Okay, so steps of transcription. So RNA polymerase and other proteins recognize the start site of a gene, okay, the start codon, a promoter, and the DNA segment begins to unwind. Okay. RNA nucleotides then form complementary base pairs with the DNA template. The growing RNA strand hangs freely as it is transcribed. As we saw in the last photo, it's hanging free willy over here, okay? The complete RNA strand separates from the DNA template and the RNA polymerase falls apart. Okay, so once it's complete, once it's all the way done, it's going to separate, and this RNA polymerase molecule here is just going to fall into pieces. It's going to disintegrate. Okay, so this is a really good animation. We are going to watch it. Okay, this is going to talk us through transcription.
Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region on the DNA, where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from free nucleoside triphosphates. The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription. Note that the bases in the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. The DNA double helix rewinds as the RNA polymerase moves through. Like the unwinding, the rewinding is an energy-requiring process that is accomplished by RNA polymerase. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely, and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.
Okay. This is a really good video to be able to come back to, so that way you can reference it if you ever get confused. Oh my God. Okay, promoters. Something we've talked about, but we haven't explained, so we're going to explain. So this promoters. How does the DNA know, RNA polymerase know where to start and where to stop making a copy of RNA, right? RNA polymerase will bind only to regions of the DNA sequence that are known as promoters. So promoters are a specific base sequence, so it's three nucleotide base pairs that indicate to the RNA polymerase where to bind. Okay, so you can see here, RNA polymerase is binding at the promoter site, and it's going to continue going. Okay, back to the central dogma. Just a reminder that we've covered the central dogma. We've covered. I am losing my mind. Replication and transcription. Okay, so genetic code. We're going to talk about the codons. So codon is a 3 nucleotide sequence that codes for an amino acid, okay? So one of these codons is going to code for AUG, and that is the start site. That is where you're going to start your code. That's where RNA polymerase binds, okay? UUC codes for phenylalanine. Codons are 3 nucleotides. 1, 2, 3. Okay, so 3 nucleotides codes for 1 amino acid. Amino acids are the molecules that make up the proteins. Okay, there are 20 different amino acids that are used to make up hundreds of combinations of proteins. The genetic code matches each RNA codon with its amino acid or function. Okay, so this is a really easy image to use to be able to identify what 3 pairs, okay, what 3 base pairs will form what codon will give you what amino acid. So if I want to start with C, let's say I want to go CCC. Okay, C to C to C gives me PRO. PRO is an amino acid. These are all very much abbreviated amino acids. Okay, and then if we were to go over here, it would be UAA or UAG is the stop. You can also go UGA, and that's another stop codon. So it's telling RNA polymerase freeze, no more. You don't need anything else. You're done. Okay. So start and stop codons. I've talked a little bit about them, but a codon can also code for signaling the end of the amino acid chain for that specific protein, or signaling the start of the site of translation and the amino acid methionine. Okay, so we go back here, AUG, methionine. Okay, there are 3 different possible ways to get a stop codon: UAA, UAG, or UGA. Okay. So the universal genetic code. For the mRNA code to be translated correctly, codons must be read in order. A change in that order, the codons are read, can change the resulting protein that is made, which would be really devastating for different functions, different gene expressions. It could even change the color of your eyes, whether or not you can see. Like it can be really devastating. Okay. This genetic code, so this fact that it needs to be read in order, is shared by all organisms. It's called the universal genetic code. Okay. So we've talked about DNA replication. We've talked about transcription. We're going to talk about translation now. I know that this has been a really bulky module. I want you to get up, take a little break, take a little second. I'm going to take a second to get a drink. In that time, just take a breather. Take a second to relax. Your hand probably hurts by now. We've been going through a lot. Okay. So translation. Translation is a process that converts or translates an mRNA message into proteins. So up until now, we've replicated our DNA so that way we can divide and we have the same amount of DNA in each cell. We've transcribed our DNA into RNA so that way we can exit and be where we need to be. We can exit the nucleus and be where we need to be. Now we need to translate our transcription. Say that 3 times fast. Okay. So that way we can make our protein. Okay. This is a really blurry image, but I still really like it just because of the fact that it is very simple. So the codon is recognized. Okay. So the 3 base pair sequence is recognized. Okay. And then the amino acids are bonded together. Peptide bond forms. Okay. And then it's shifted. It keeps getting shifted further and further so that way more amino acids can bond and the amino acid chains get longer and longer and longer. Okay. So ribosomes. We've talked a little bit about ribosomes. We're going to talk a little bit more. Ribosomes are made up of 2 large subunits. Okay. So this top subunit is known as the large subunit. It has 3 binding sites for tRNA. Remember that funky looking molecule I showed you earlier? Inside the ribosome, the tRNA is going to come in. It's going to sit in one of the binding sites. Okay. And then it's going to use the small subunit to bind to the mRNA. Okay. And that will what produces the amino acids. Okay. So ribosomes are made up of rRNA and proteins. Okay. tRNA. Okay. So this is the funky looking R. Okay. It acts as an adapter between mRNA and amino acids. Okay. One end of the rRNA molecule has a specific amino acid. Okay. So the small subunit has a specific amino acid sequence, right? The other end, okay, of the tRNA recognizes that specific codon. So it recognizes it here, and then it goes up and sends and anticodon. So 3 different complementary nucleotides go up and pop off an amino acid, okay? So the steps of translation. 1, the exposed codon attracts tRNA molecules carrying an amino acid. The tRNA anticodon pairs with the mRNA codon, bringing it very close to the other tRNA molecule. The ribosome helps form a peptide bond between the 2 amino acids, while the ribosome breaks the bond between the tRNA in the second site and its amino acid, okay? So it's breaking the bond between the tRNA and the amino acid, and it's forming a bond between this amino acid and another amino acid. So it's starting to form an amino acid chain, okay? The ribosome pulls the mRNA strand to the length of the code of 1 codon, okay? 1 set of 3 base pairs. The tRNA molecule in the second site is shifted to the third site. So it's just, eventually, once it gets past the third site, the tRNA leaves the ribosome to get another amino acid, okay? So as you can see, incoming tRNA is going to be complementary to this, okay? It's going to bump this tRNA molecule out so it can go find another amino acid and come back, and it just repeats as a cycle, okay? And you can see that these bonds are forming while these bonds are breaking. So that was module 3. Take a little break, take a breather. If you're doing all this in one night, please eat some food. If you're doing this the night before the test, please eat something. Please, 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 okay? But this is module 4. We're not going to go into module 4 right now. Please go into the DNA invitation notes. That way you know exactly what you need to complete for module 4. Have a great time studying for your test, and I will catch you on the flip side.